Hi, I'm Deeksha and this video will be talking about malaria. The pictures and stuff you see here are what I used for a pediatric presentation on malaria, and that's what I'll be using for the video today. We'll talk about etiology, life cycle, pathogenesis and clinical features in this video. We'll cover up diagnosis, treatment, management, prevention aspects in later video sometime later. Okay, starting with malaria. Epidemiology. The regions which are mostly affected with malaria are the tropical, subtropical areas, as we see here, this region of the world, and it's endemic in all of these places. Africa is highly affected with malaria. As we can see, India is also endemic to malaria, and that's why we need to know it's a common disease, and we have the vector present, and we have the agent present, the Plasmodium. So we need to know how to tackle all of this. That's why we are learning all of this. Agent, host, environment triad, as we say. The agent is Plasmodium. Plasmodium is a parasite that we all know. It has 5 species: Plasmodium falciparum, vivax, ovale, malariae, and latestly the known found out one is the Naulesi. Okay, these are the 5 species of Plasmodium parasite we know about. And then host factors. Host, who are the people who are susceptible to the disease? Travelers to and from an endemic area are susceptible to the disease. What do we mean by to an endemic area? So the person is not exposed to the disease in his original place, and when he goes to an endemic area, he's more prone to get infected by the infected and get the disease. Okay, from an endemic area, the person can transmit this disease to the people to the region he's going to. Okay, so that's what we call as travelers to and from endemic areas. Children, pregnant women, these category of people are highly prone to get a severe form of malaria. Latent infection. If the person is living in an endemic area, he's exposed to the parasite, so he'll have some immunity to it. But then he has an infection but may not necessarily develop the disease, right? So that's called latent infection. Premonition. What is this? When this person from this endemic area goes to another area which is not endemic, so he loses the immunity that he developed here. And that is called as premonition, meaning the loss of immunity when the person moves from one region which is endemic to a region which is not endemic. Okay, the host factors. Vector, we know it's a female Anopheles mosquito which transmits this Plasmodium from one person to another person. Environment is related to this vector. The vector breeding sites are places where this disease exists. So clean, slow water paddy fields are where this vector prefers to breed. Okay, so these are the environment factors of this disease. Now, little bit mode of transmission, it's mostly vector borne. This female Anopheles mosquito transmits it from one person to the other person. It can happen vertically as well, meaning mother to fetus through the placenta. If there is any placental defects, the parasite can enter from the mother to the fetus through the defective placenta. Blood. If there's if infected blood products are transfused to another person, the person can get the disease. Okay, so blood should be infected. Next we'll talk about the life cycle of the parasite. Okay, we see here the definitive host is the mosquito, meaning the sexual cycle of the parasite takes place in the mosquito. The intermediate host is the human, meaning the non-sexual cycle of the parasite, asexual cycle of the parasite takes place in the human. Okay, intermediate host is human. How does a human get infected? The sporozoites is the infective form of the parasite which stays in the mosquito's salivary gland. Okay, these, when the mosquito bites the person, these enter the blood of the human, and within 30 minutes or so it enters the liver and enters the liver cells. Okay, and in the liver it develops various phases of it develops into various forms. It infects the liver cell and then it forms schizons in the liver cell, and these schizons further rupture, and what they release is called the hepatic merozoites. These hepatic merozoites then enter the blood. in the blood they enter the RBCs. Okay, in the RBCs they again undergo further development. They form trophozoite, the ring form of the trophozoite, and then further schizons are formed, and then all these are like multiplicative stages. Okay, all these multiply, and then one point of time the RBC which is infected and has formed schizons ruptures, releasing merozoites, okay, into the bloodstream, and this rupture is what is associated with causing fever. When the RBC ruptures, it releases merozoites. These merozoites are responsible. This rupture, this hemolysis and release of merozoites corresponds to the episode of fever the person gets. Okay, this is the one part of the cycle. And RBC stage also produces gametocytes, and these gametocytes are the infective form to the mosquito. So when the mosquito bites an infected person, it takes the gametocytes, and these gametocytes fertilize in the mosquito, and they form ookinete, oocyst, all of these, and then finally form sporozoites which stays in the salivary gland of the mosquito, and then it bites another person, and this is how the whole cycle continues. Okay, and we have two terms that we have to learn here. One is relapse, one is recrudescence. What is relapse? We saw the parasite has a liver stage. In the liver stage, it forms schizons, and then it forms these schizons further ultimately form merozoites, which are entering the bloodstream. But in cases of Plasmodium vivax and Plasmodium vivax especially, these schizons can form dormant phases. Then they can have a dormancy in the liver, meaning they just stay in the liver without multiplication, and these stages are called hypnozoites. These hypnozoites are what are responsible for relapse of the disease, meaning these hypnozoites are remain dormant in the liver, and sometime later on in life, these hypnozoites can get activated. They were essentially schizons, right? They can get reactivated, and they can form merozoites which are released into the bloodstream. So these are like dormant phases which can reactivate later, and that's called relapse of the disease. Okay, and then we have something called recrudescence. What is recrudescence? It mostly occurs in Plasmodium falciparum. What happens here is, if the person has developed malaria and is not adequately treated, and he can develop the disease later on. Okay, what do we mean by inadequately treated? He could be drug resistant, he could have received inadequate doses of the drug, he could have not been compliant to the drug, he would have not taken the drug properly, all of these. Okay, all of these could be various reasons which could cause the parasite to stay in the RBC, so meaning the blood is not fully cleared of the parasite. So these parasites which remain in the blood without being destroyed even after the medication has all been taken, that is called, and this can cause disease later, that is called recrudescence. How do we differentiate relapse and recrudescence? With the cutoff range of 28 days. If the person he develops the disease, he had malaria, and then he develops another episode of malaria at less than 28 days, it's called recrudescence. Okay, if he develops the disease again after later than 28 days, it's called a relapse. So this 28 days is the cutoff for relapse and recrudescence that we talk about. Then we'll talk about pathogenesis of the disease. So here we need to know that erythrocytic phase of the RBC phase is the pathogenic phase. The parasites in the RBCs are what is responsible to cause the disease. The liver stage is the extra erythrocytic stage of the cycle, has no role in causing the manifestations of the disease. Okay, the RBC stage causes the manifestations of the disease. What are all the manifestations? What is the pathogenesis that it can cause to? Infected RBCs, parasites which infect the RBCs, right? The RBCs which are infected, they adhere to the blood vessels, they can obstruct blood flow. We'll talk about adherence to blood vessels mostly in Plasmodium falciparum. We'll come back to it. Okay, the RBCs are infected, phagocytosis of the RBCs occur. The body is trying to defend itself, so it phagocytoses the RBCs, where there's be hemolysis, rupture of the RBCs which are infected, that can cause anemia. Then there's extensive hemolysis, so look, the kidney is trying to throw out all the waste products which are formed, so there can be renal failure, kidney injury. That there's hemoglobinuria, that is called black water fever. Okay, and there can be splenomegaly, hepatomegaly, because these are the organs which are essentially trying to phagocytose and throw out, sequester all the infected RBCs. So these there will be compensatory enlargement of all these organs. So that is splenomegaly, hepatomegaly. And then it's the toxic mediators, the inflammatory substances, inflammatory responses are produced, and metabolic disturbances. The inflammatory responses are responsible for fever. We know malaria is a febrile illness. There will be chills, fever, sweat. This is like the stages of the disease that the person goes through. We'll talk about this in clinical features. Okay. Talk about adherence. I spoke about adherence to blood vessels, which obstructs blood flow. This obstruction to blood flow causes tissue hypoxia and impaired microcirculation. These can lead to end organ damage. The brain can be affected, causing cerebral malaria. There can be pulmonary edema or pulmonary superadded bacterial infection can occur, and all the blood trying to compensate. There can be DIC and all of those. Okay, Plasmodium falciparum infection is where we talk about adhesion of RBCs to the endothelial cells. These obstruction to blood flow we talk about in Plasmodium falciparum infection. This Plasmodium falciparum is responsible to cause a very severe form of malaria. Why so? Because of this substance called as Plasmodium falciparum erythrocyte membrane protein 1, which is present on the RBC is infected with this Plasmodium falciparum parasite. Okay, what does this do? When the RBC which has Plasmodium falciparum infection, this has this receptor, it passes through the blood vessel, endothelial lining, blood vessel, right? So that has endothelial lining. These receptors attach to the endothelial lining, so essentially they attach to it and they don't flow, they don't move through the blood vessel. So all of these lead to blockage of the vessel. So there's more RBCs which are getting adhered to the blood vessel, and there's blockage of all the vessels with the RBCs, and essentially this causes microvascular circulation impairment and tissue hypoxia. So that's why the end organ damage what we talk about. So that is responsible for the severe form of malaria and Plasmodium falciparum. Protective factors, we know there are certain protective factors to this disease, meaning the person with all of these stuff, all of these things cannot get the disease, or gets a milder form of the disease. Is sickle cell trait. In sickle cell trait, there is abnormal shape of the RBCs. We know there's these abnormal sickle shape cells undergo hemolysis. So the parasite essentially cannot survive in an abnormal RBC. Okay, and then Duffy negative blood group. In Plasmodium vivax it is significant, where Plasmodium vivax needs the Duffy receptor on the RBC to enter the cell. If this receptor is not present, that means the parasite cannot enter the cell. G6PD deficiency, G6PD deficiency. We know G6PD deficiency tackles oxidative stresses in the cell. When that is deficient, meaning the RBC has excess amount of reactive oxygen species, and the parasite cannot survive, cannot multiply, cannot survive in a red cell which is oxidatively stressed. It needs a healthy red cell to survive. So all these factors are protective. It prevents the person from getting the infection. We will now talk about clinical features of malaria. The classic triad is anemia, splenomegaly, fever. Anemia because of all the hemolysis, which is of normocytic hypochromic type. Fever, it's a febrile illness. There's all the inflammatory processes which are occurring which causes fever. There's splenomegaly because of all the sequestration and the body trying to sequester all the RBCs. The spleen gets enlarged. That is splenomegaly. So this is the triad that's always present. Okay, we'll talk about fever in detail. The fever pattern is called intermittent fever. What do we mean by intermittent fever? Meaning the fever touches meaning the fever touches the baseline and there's a spike of fever, it stays, and then it touches the baseline again, it goes up, fever spikes, and then touches the baseline, and then there's a spike again. And this spike, this difference is more than 1 degree Celsius. Okay, so this is the intermittent sort of fever that is classic to malaria. How is this? There's a afebrile phase, there's a febrile phase, another afebrile phase. This is how the fever continues. Intermittent fever. Okay, febrile phase has 3 phases, 3 stages of the fever. There's a cold stage, there's a hot stage, there's a sweating stage. What do we mean by cold stage? The person starts feeling chills and rigors in the cold stage, and then that lasts for some amount of time. And then there's a hot stage where the person is like has like very high temperature and he's looking all reddish and flushed. That is the hot stage. And there's a sweating stage where the person has excessive sweating, there's excessive perspiration, and then there's fall in temperature. So and the person becomes afebrile again. And after this afebrile period, when the febrile period occurs, all these stages again continue. So this is intermittent character or the fever that we talk about. And this intermittent fever is different in different species of the parasites. And in Plasmodium falciparum, it is ideally supposed to be tertian fever, meaning the fever occurs every 48 hours. Okay, meaning the fever comes on first day, and then on the third day again it comes. So meaning the fever comes on alternate days. First day it comes and then third day it comes. This thing is variable in Plasmodium falciparum. If the person is infected with this, we do not essentially see a tertian form of fever type. It's mostly quotidian fever type, or it can be variable, it can be anything. Okay, so that it's not specific to Plasmodium falciparum. And it's also called malignant tertian because in falciparum the disease is severe. Okay, then we have Plasmodium vivax and oval, which also have a tertian pattern of fever. First day fever occurs and third day fever occurs every 48 hours. Okay, then we have Malaria, which has quartan kind of fever, which is first day fever occurs and fourth day fever occurs. So every 72 hours fever recurs, the intermittent pattern of fever. Naulacea has a quotidian kind of fever, meaning every 24 hours, every next day the fever comes. Every 24 hours the fever recurs. This is the intermittent pattern of fever that we talk about in different species of the parasite. Then we'll talk about the other clinical features that can occur. We spoke about the triad, which is anemia, splenomegaly, and fever. Then there can be end organ damage, I spoke about. There can be cerebral malaria, where the person can have coma, hypoglycemia, seizures, and all of these. The child can throw up a seizure, the child can become unconscious, the child can have reduced feeding. All of these are like a child can present with, a neonate or a kid can present with. Okay, then there can be respiratory diseases, respiratory damage, edema, bacterial pneumonia, superadded infection can occur. There is extensive hemolysis, the person can have reduced blood volume, the person can have a cardiogenic shock, a cardiac failure can occur, which is called algid malaria. Okay, and then there can be renal injury, where because of all the hemolysis, kidney injury can occur. That is called black water fever, where the urine is dark colored. And abdominal jaundice can occur because of the hemolysis and extensive bilirubin, which is formed out of the ruptured RBCs. There can be jaundice, and tender hepatomegaly and splenomegaly can also. Then we'll talk about the differences between the various species of the parasite. Incubation period is like pretty much the same, but it can be variable across all the species. Hypnozoites, we spoke about hypnozoites, the dormant stages in the liver. So these are present in vivax and they're present in oval. What do they have affinity for? By affinity I mean what the liver merozoite which is released, which kind of RBC does it affect? In Plasmodium vivax it affects the young RBCs. In Plasmodium malaria, it affects the older RBCs. In Plasmodium falciparum, it is very non-specific, meaning it affects both young and it affects both old RBCs. So that's why it affects all the RBCs. That's why the disease is more severe in case of a Plasmodium falciparum infection. Then fever periodicity, we saw through this chart here, this graph here. vivax every second day, meaning every 48 hours. Plasmodium ovale also every second day. Then Plasmodium malaria every third day, and then Plasmodium falciparum also every second day, but this is variable, right? And then relapse because of the hypnozoites is present in Plasmodium vivax, Plasmodium ovale. Recrudescence we saw is a feature of inadequate treatment, especially in Plasmodium falciparum, and also called these diseases are also called as by these names. You need to remember malignant tertian malaria in Plasmodium falciparum, malignant because it's very severe. So this is what we saw today. We spoke about epidemiology, the agent host, the environment factors of malaria, the life cycle of the malarial parasite, the intermediate host being human, the definitive host being mosquito. Sporozoid is the infective stage of the parasite, gametocyte. Sporozoid infective stage to the human, gametocyte is the infective stage to the mosquito, and the erythrocytic cycle is what is responsible for the manifestations of the disease. We saw the difference between relapse and we saw the recrudescence. Then we saw what the pathogenesis of the disease, of some protective factors for the disease, and why Plasmodium falciparum infection is very severe. And we saw the triad of clinical features, and we saw the types of intermittent fever which is there in malaria, and few important differences between the parasite. And that's all for today. Thank you so much. Bye.